Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
All right, let's talk about something huge, something affecting maybe 1 in 3 adults right here in the US. It's often silent, chronic, fatty liver disease. You might hear it called MASLD now, metabolic dysfunction-associated steatotic liver disease. Or its close cousin, alcoholic liver disease, driven by alcohol. And look, this isn't just a minor issue. It's a serious public health problem. It can lead down some really difficult roads: type 2 diabetes, cirrhosis, even liver cancer. For a long time, the basic idea seemed simple enough. Too much fat just builds up in the liver cells, usually because of metabolic stress, diet, obesity, or from chronic heavy drinking. But why exactly? What's the precise molecular switch that flips what breaks down inside the cell? That part has been, well, kind of fuzzy. Until now. Today, we're doing a deep dive into a really significant discovery. Researchers at Mayo Clinic, writing in the Journal of Cell Biology, they've actually pinpointed the exact molecular steps, the mechanism for how chronic alcohol exposure completely messes up the liver system for managing fat. So our mission here is to basically shortcut you right to the heart of it. We want you to understand these critical cellular mechanics. We'll name the proteins, talk about the cleanup crew inside the cell, and show you exactly how alcohol seems to just shut down this vital recycling operation. And then, of course, we'll get into what this incredibly detailed picture could mean for new treatments down the line.
Yeah, this really is a foundational piece of work. It moves us beyond just observing the condition. It gives us a specific molecular target, something potentially actionable inside the liver cell.
Yeah.
But before we get into the breakdown, we really need to appreciate what the liver does. It's the body's main processing plant, isn't it? The primary filter for almost everything.
Absolutely. And the workhorses doing all that filtering are the liver cells themselves, the hepatocytes?
Exactly. Hepatocytes perform this absolutely massive job. They're constantly taking in stuff from our gut, sorting it, detoxifying things, breaking down waste, recycling materials. It's nonstop. And a key part of their job is managing fat.
Yep.
Fat comes in from our diet, or the liver makes some itself, and it gets stored inside these hepatocytes. It's stored in these little structures, globules really, called lipid droplets.
So like tiny fuel tanks inside the cell.
That's a great way to think about it. They're temporary energy reserves, really important if you're fasting, for example. But when these droplets start to accumulate too much, when they get too big or too numerous, well, that's hepatic steatosis. That is fatty liver disease.
Okay, let's unpack this then. The liver's supposed to be this master recycler. So what happens when the recycling machinery itself, specifically around these fat depots, just breaks down? Because that failure seems key to steatosis.
Right. And according to this Mayo Clinic study, the central breakdown point seems to involve a critical interaction, or lack thereof, between two specific proteins. The first one is, let's call it the cell's internal coordinator. It's an enzyme called the Valosin-Containing Protein, or VCP. Now, VCP isn't just in the liver. It's fundamental pretty much everywhere in the body. Its general job is as a segregase. Think of it like a specialized tool that uses energy, ATP, to literally pull protein complexes apart. It's crucial for keeping proteins in order what we call homeostasis. It recognizes damaged or misfolded proteins, especially ones stuck on membranes inside the cell, and pulls them off so they can be dealt with, usually degraded and recycled.
So VCP is like the cell's bouncer and quality control manager rolled into one, making sure damaged goods or things that don't belong are removed efficiently from where they shouldn't be.
That's a very good analogy. And in a healthy liver, VCP, our coordinator, is doing a really specific vital job right there on the surface of those lipid droplets, those fat storage tanks. Its presence there is absolutely essential for keeping the levels of a second protein in check. And this second protein seems to be the problematic one when things go wrong. It's called hydroxysteroid dehydrogenase 17 beta type 13.
Okay, whoa, that is quite a name. HSD 17 beta 13. Since this is the one that seems to pile up and cause trouble, maybe we can call it the accumulator, just keep track.
The accumulator, I like it. So HSD 1713, the accumulator, it actually lives right there on the surface of the lipid droplet. And the problem is when too much of the accumulator builds up on that surface, the amount of fat stored inside the liver cell just balloons. This directly contributes to the steatosis. Now, in a healthy liver cell, VCP, the coordinator, is right there on the droplet surface too, actively removing the accumulator, HSD 1713, keeping its levels low and preventing that harmful buildup.
Okay, the picture's getting clearer. VCP keeps HSD 1713 in check on the fat droplet. So now, the million-dollar question. How does chronic alcohol exposure completely wreck this balance? How does it break that VCP-HSD1713 connection? The researchers started by looking for correlations in animal models. They took rats and fed one group a liquid diet with about 30% ethanol for 8 weeks. That's, modeling chronic heavy alcohol intake. They compared them to a control group on a normal diet.
Yeah.
The next step was really specific. They actually isolated the lipid droplets themselves from the livers of these rats. Then they used a powerful technique, mass spectrometry, to basically take inventory to see exactly which proteins were present on the droplet surface and how much of each.
And the findings were, well, pretty striking. They weren't subtle changes, they quantified it. The amount of VCP the coordinator found on the lipid droplet surface, it showed a huge drop. We're talking more than a 10-fold decrease in the alcohol-fed rats compared to the controls.
Wow, tenfold. That's basically gone. It's like the coordinator was almost completely kicked off the job site, right where the fat regulation is happening.
It's a near-complete functional eviction, exactly. It strongly suggests that chronic alcohol exposure effectively removes this protective cleanup coordinator from the fat depots. And maybe, predictably, the consequence was perfectly reciprocal. When VCP disappeared, the levels of HSD1713, the accumulator, shot up. They measured a substantial 7-fold increase in the accumulator protein sticking to those same lipid droplets.
So the bouncer leaves and the problematic stuff just rushes in and takes over.
That's exactly the picture the mass spectrometry painted. Alcohol seems to cause VCP loss, which then directly leads to this HSD1713 accumulation. And we already know that accumulation is linked to the fat buildup, the steatosis. And importantly, this wasn't just seen in the rats. The researchers also looked at liver tissue samples from human patients, people suffering from severe alcohol-associated steatohepatitis, which is fatty liver with inflammation. And they observed significantly increased staining for HSD1713 in those human samples too.
Okay, seeing it in rats and humans makes the correlation much stronger. But as always in science, correlation isn't quite causation. So the next big step for this team was to prove definitively that losing VCP itself actually causes the fat buildup, even without any alcohol involved. Right? They needed to isolate VCP's role. And they tackled this with, I think, 3 really clever experiments. First, they worked with mouse liver cells, hepatocytes, in a dish. They used something called siRNA, small interfering RNA. It's like a molecular tool to specifically silence or turn down the gene for VCP. So what happened when they just reduced VCP levels artificially?
Well, the fat management system went haywire, just as the correlation suggested. Simply reducing the amount of VCP protein in the cells led to a 3- to 5-fold increase in the total amount of lipid droplets measured by area. So less VCP meant more fat storage.
Okay, so VCP's presence is clearly needed, but is it just sitting there, or does its activity matter?
Great question. That's what the second experiment addressed. They used a chemical, a drug called DBQ. Now, DBQ is known to specifically inhibit the enzymatic activity of VCP. Remember, VCP uses energy, ATP, to pull proteins apart. DBQ blocks that energy-using function. And when they treated the cells with DBQ, they saw a clear dose-dependent increase in lipid droplet content. The more they inhibited VCP's activity, the more fat built up. This was crucial because it proved VCP isn't just structurally important. Its active job, its segregase function, is essential for controlling fat levels.
That makes perfect sense. You don't just need the coordinator present, you need the coordinator actively working. But the real clincher, the definitive proof, often comes from moving this into a living animal model, right? Taking it beyond the petri dish.
Absolutely. And that was the third and perhaps most powerful piece of evidence. They developed a conditional liver-specific VCP knockout mouse. It's quite sophisticated. They used special viral vectors, AAV8-BG-CRE, designed to deliver genetic instructions that would effectively delete the VCP gene only in the liver cells of the mouse.
Only in the liver. That's key, so you're not messing up VCP's function everywhere else in the body.
Exactly. It isolates the effect to the organ we're interested in. And these mice, importantly, were just kept on a standard healthy chow diet. No alcohol involved here.
And the result of just deleting VCP in the liver of these otherwise healthy mice.
It's dramatic. Within a remarkably short time, just 7 to 13 days, these mice developed severe fatty liver. They showed a greater than 10-fold accumulation of lipid droplets and triglycerides in their liver tissue compared to control mice that still had VCP.
Tenfold again, just by removing VCP. That's astounding. It happens that fast, even on a normal diet. It completely nails down the idea that VCP is acting as a critical gatekeeper, actively protecting the liver against fat accumulation.
Okay, so the case is incredibly strong now. VCP is essential. Alcohol makes it vanish from the lipid droplet. When VCP is gone, the accumulator, HSD1713, piles up, and that leads to steatosis. The next really deep question then becomes, how does VCP normally get rid of HSD1713? When it pulls it off the droplet, where does it send it for disposal? The researchers' first guess, based on VCP's known roles elsewhere, was probably the proteasome. That's the cell's main protein shredder, the typical destination for misfolded proteins that VCP handles. But here's where the story took a really interesting turn.
Yes, this was a surprising result that really changed their thinking about VCP's function in this specific context. They treated the liver cells with the standard drug, MG132, which is known to block the proteasome.
Yeah.
If HSD1713 was being sent there, blocking the shredder should cause it to pile up, right?
Makes sense. But it didn't. Treating the cells with a proteasome inhibitor, MG132, had basically no effect on the levels of HSD1713.
Yeah.
The accumulator levels stayed the same. This was strong evidence that, in this case, the proteasome wasn't the disposal route. So if not the proteasome, where else could it go? They then tried inhibiting a different cellular disposal system, the lysosome. The lysosome is more like the cell's recycling center. They used a drug called chloroquine, or CQ, which blocks lysosomal function.
And what happened then?
The accumulator levels shot up dramatically. Treating the cells with chloroquine caused HSD1713 levels to increase somewhere between 5 and 8-fold. This immediately suggested that the lysosome, not the proteasome, was the actual destination where HSD1713 was being broken down.
Hold on. So VCP, which is famous for sending proteins to the proteasome, is instead directing this specific protein, HSD1713, to the lysosome. That seems like a completely different pathway. How does VCP even do that? How does it get the accumulator from the fat droplet over to the lysosome?
That's precisely the next mechanism they unraveled. It turns out VCP uses a very specific, highly regulated pathway to the lysosome called chaperone-mediated autophagy, or CMA. CMA isn't like general autophagy where big chunks of cytoplasm get engulfed. It's much more selective. And in the CMA pathway, VCP seems to be acting as a co-chaperone. It doesn't do the whole delivery job itself. It partners up with another key protein, a cytoplasmic chaperone called HSC70.
Okay, so VCP is the coordinator, and now we have HSC70, which we can maybe call the guide.
The guide. Yes, that fits perfectly. HSC70, the guide, is the protein that actually recognizes specific cargo proteins that are marked for this CMA delivery route to the lysosome. It looks for a particular sequence of amino acids on the target protein, like a shipping label. This is known as the KFE or Q motif. And the researchers actually identified this specific shipping label sequence within the accumulator protein, HSD1713. It's the amino acid sequence 275-ERS-279. That's the signal that tells HSC70, okay, take this one to the lysosome via CMA. So the model is VCP first pulls HSD1713 off the lipid droplet surface. Then acting as a co-chaperone, it likely helps present HSD1713 to HSC70, the guide. HSC70 recognizes that HSC sequence, binds to HSD1713, and escorts it directly to the surface of the lysosome. There, the whole complex interacts with another protein embedded in the lysosome membrane called LAMMP2A. LAMMP2A forms a kind of pore or channel, allowing HSC70 to thread HSD1713 directly into the lysosome, where it's finally broken down by the lysosomal enzymes.
That is an incredibly detailed pathway they've mapped out. From VCP, pulling it off the droplet, handing it to HSC70, identifying the specific sequence signal, escorting it to the LAMP2A pore and into the lysosome. Did they have evidence for these specific steps, like the LAMP2A pore being necessary?
They did. They performed another knockdown experiment, this time targeting the LAMP2A pore protein. When they reduced the amount of LAMP2A, the levels of HSD1713, the accumulator, increased by over 50%. This confirmed that this specific CMA channel is crucial for clearing HSD1713. And critically, they also showed the functional link between VCP and HSC70 in this process. Remember the VCP inhibitor drug, DBQ. When they treated cells with DBQ to block VCP's activity, they measured the physical interaction between HSC70, the guide, and HSC1713, the accumulator. They found that inhibiting VCP reduced that essential interaction by about 60 to 70%. This suggests that VCP's activity is somehow required for HSC70 to properly recognize or bind to HSC1713, perhaps by correctly unfolding or presenting that KVRQ-like signal. The coordinator needs to rep the package for the guide.
Okay, this really ties the whole disruption story together then. It's a cascade failure initiated by alcohol. Let's recap. In a healthy liver cell, VCP, the coordinator, is anchored to the lipid droplet. It's actively working, likely with HSC70, the guide, to recognize and pull off excess HSD1713, the accumulator. It then directs it via that specific CMA pathway using the ERSA signal and the LAMP2A pore into the lysosome for destruction. This keeps fat levels stable. but introduce chronic excessive alcohol. This causes VCP to detach almost completely, that huge tenfold loss from the lipid droplet surface. And if VCP isn't physically there, anchored on the droplet, it simply cannot perform its job. It can't grab HSC1713, it can't act as the co-chaperone to hand it off to HSC70 for CMA degradation. So the accumulator, HSC1713, just piles up relentlessly on the lipid droplet surface. This buildup somehow disrupts normal fat metabolism. The exact downstream effect is still being studied, but the result is clear. The fat content skyrockets and you get steatosis. The whole cleanup system fails right at that initial VCP anchor point.
Exactly. And the clarity of this step-by-step mechanism is why it's potentially so powerful for developing new therapies. Dr. Mark McNiven, the senior author on the study, specifically highlighted this therapeutic potential. Because we now have names and functions for the key players in this failure, VCP, HST1713, HSC70, LAMP2A, it opens up multiple potential points for intervention. Maybe we can develop drugs that help stabilize VCP on the droplet surface, even in the presence of alcohol stress. Or perhaps we could target HST1713 itself, either blocking its accumulation or maybe enhancing its degradation through the CMA pathway, even if VCP levels are low.
Yeah, I can see that. Maybe a future therapy wouldn't even need to directly boost VCP. What if you could just make that LAMP2A pore work more efficiently, or somehow enhance the ability of HSC70, the guide, to find and grab the accumulator? You'd essentially be boosting the downstream recycling capacity to compensate for the upstream VCP loss.
That's precisely the kind of thinking this detailed mechanism enables. And beyond treatment, there's also potential for prediction. If we could develop ways to measure how efficiently this VCP HSD 1713 cleanup system is working in an individual, maybe by looking at protein levels or interactions in blood samples or biopsies, we might be able to identify patients who are inherently more vulnerable to developing severe fatty liver disease if they consume excessive alcohol. Perhaps their baseline VCP function or CMA capacity is lower to begin with.
That ties into the bigger picture, too. This research, as mentioned, falls under Mayo Clinic's Procure initiative. The whole goal there is to find ways to spot these kinds of biological processes going wrong before they snowball into full-blown, hard-to-treat diseases. They want to intercept the problem at the molecular root, like this VCP detachment. Now, we absolutely have to address something important here for a complete picture. There's been some confusing or conflicting data out there about HSD1713, the accumulator itself. It's not universally seen as purely detrimental in all contexts, right?
That's a crucial point of nuance, yes. While this study very clearly shows that HSD1713 accumulation driven by VCP loss leads to steatosis in their models, the broader literature on HSD1713's intrinsic function is, well, complicated. For instance, there are studies in humans looking at genetic variations. People who naturally have certain genetic variants that result in a less active or loss-of-function version of the HSD1713 protein, these individuals actually seem to be protected against the progression of chronic liver diseases, including steatosis progressing to fibrosis or cirrhosis.
So having less functional HSD1713 seems good in humans, but this study shows its accumulation is bad. And then, didn't some earlier lab studies, maybe in cell cultures or different mouse models, show conflicting results when they tried to increase or decrease HSD1713 levels directly? Sometimes fat went up, sometimes down, sometimes no change.
Exactly. It's been inconsistent, leading to this puzzle. Why the discrepancy? Is HSD1713 good or bad? Does the context alcohol versus metabolic dysfunction, mouse versus human, change its role dramatically? This new VCP discovery offers a potentially elegant way to reconcile some of this. Perhaps the critical issue isn't solely about HSD1713's inherent enzymatic activity, what it does chemically. Perhaps the primary problem, at least in the context of alcohol-induced VCP loss, is simply its physical accumulation on the lip droplet surface.
So maybe just having too much of the protein physically stuck there gums up the works, interferes with other fat-regulating enzymes on the droplet, regardless of whether that specific HSC1713 molecule is actively doing its own enzymatic job or not.
That's a strong possibility raised by this work. The location and clearance might be more important than the intrinsic function in this specific disease pathway. The VCP mechanism focuses entirely on regulating the amount of HSD1713 on the droplet via CMA degradation. And let's loop back for a second to the very beginning of the disruption, how alcohol causes VCP to detach. We said VCP is like a multi-tool, and it often needs specific adapter proteins to connect it to different places in the cell, like different membranes. Well, when the researchers did their detailed mass spectrometry analysis of the lipid droplets from those alcohol-fed rats, they didn't just see the massive loss of VCP itself. They also noticed a significant concurrent loss of a specific VCP adapter protein called UBXD8.
Okay, wait. So maybe alcohol isn't directly kicking BCP off the droplet. Maybe it's actually damaging or moving UBXD8, the docking station that VCP needs to attach there in the first place.
That's exactly what the data suggests. And they tested this too. They experimentally messed with UBXD8 function. They introduced just a tiny mutation, changing only 3 amino acids in the part of UBXD8 that normally binds to VCP. And that small change was enough to prevent VCP from properly targeting and anchoring onto the lipid droplet surface. And the result, just like removing VCP directly, disrupting the UBXD8 adapter led to an increase in fat accumulation in lipid droplet content. So this adds another layer. Alcohol might initiate the whole cascade by disrupting this UBXD8 adapter, which then prevents VCP from docking onto the fat droplet, which then prevents HSD1713 clearance via CMA, leading to its accumulation and ultimately steatosis.
It really highlights how interconnected these cellular systems are. A failure in one small connecting piece can bring down the whole regulatory pathway. It also suggests that maybe stabilizing that VCP-UBXD8 interaction could be another therapeutic angle.
Potentially, yes. It gives us more specific targets to consider.
This has been a fascinating journey deep inside the liver cell. It really drives home how a major health problem like fatty liver disease ultimately stems from these very specific, reproducible molecular failures. So let's try to crystallize the main takeaway here. What this research reveals is that chronic, heavy alcohol consumption triggers a critical failure. It removes a vital cellular coordinator, VCP, from the surface of fat storage droplets in the liver. This removal is likely due to disruption of its anchor protein, UBXD8. Once VCP is gone, it can no longer perform its newly discovered role as a co-chaperone for a specific recycling pathway called chaperone-mediated autophagy, CMA. This means it can't guide another protein, HSD1713, the accumulator, which promotes fat storage, towards the lysosome for destruction.
That's right. The accumulator then piles up on the fat droplet surface, disrupts the normal balance of fat metabolism, effectively locking the fat inside the cell, and driving the development of steatosis or fatty liver. The real significance here isn't just identifying these proteins, but mapping out this unexpected CMA degradation route coordinated by VCP. It gives scientists and drug developers a much more precise molecular handle on the problem, moving beyond general ideas about fat buildup to a specific, targetable cleanup pathway that fails. And maybe, just to leave you with a final thought, let's connect this back to the bigger picture again. We know VCP is crucial for maintaining protein quality control throughout the cell, not just for HSD1713 on lipid droplets. This study focused on alcohol-related liver disease. But if chronic alcohol consumption can cause VCP to detach from membranes and disrupt this vital CMA recycling pathway in the liver, it forces us to ask, what other essential cellular cleanup and maintenance mechanisms might be similarly compromised, perhaps more subtly, across the entire body by similar levels of alcohol exposure? Could this insight into liver steatosis actually unlock a more fundamental understanding of how chronic alcohol impacts the body's overall capacity for cellular repair and maintenance? This very specific finding might have much broader implications for understanding alcohol's systemic effects.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.